I'm Daniel Levine, and this is the Bio Report.
Antibody-drug conjugates have established themselves as an important cancer treatment modality, but their effectiveness and tolerability in solid tumors can be constrained by inadequate tumor delivery, incomplete internalization, and off-target toxicity. NEOK Bio is seeking to overcome those challenges with its bispecific ADC platform that uses dual-antigen targeting, engineered antibodies, and next-generation linker technology. Mayank Gandhi, co-founder and CEO of NEOK, discusses the company's bispecific ADC platform, its 2 clinical programs, and its strategy to develop more selective therapies for patients with advanced solid tumors. Mayank, thanks for joining us.
Happy to be here. Thanks for having me.
We're going to talk about bispecific antibody-drug conjugates, NEOK Bio, and its efforts to overcome the limitations of first-generation antibody-drug conjugates. Let's start with those, though. For listeners not familiar with ADCs, what are they and how do they work?
So ADC stands for antibody-drug conjugate, which essentially is an antibody-based therapy, as the name implies. So you have an antibody that is targeting an antigen or a set of antigens typically expressed on tumors. That is sort of a homing device, if you will. And linked to that antibody is a payload, typically a toxic payload, a chemotherapeutic payload, attached to the antibody via a linker. That can be a cleavable linker or a non-cleavable linker, but those are details. So antibody-drug conjugates basically have 3 key components. You have the antibody, you have the linker, and you have the payload, all attached together to form this one construct called an ADC.
And what have been the limitations of these therapies, particularly with regards to treating solid tumors?
ADCs have been a proven modality at this point. There are several ADCs already approved, commercially available for the treatment of a variety of tumors, primarily hematological cancers and increasingly solid tumors. However, not many ADCs have had success, or ADCs have had limited success in the vast majority of solid tumors for a variety of reasons. I would consider the key limitations of conventional ADCs that revolve around both efficacy and safety. So one of the key ways to generate activity and efficacy for an ADC is what we call internalization. So once an ADC reaches the tumor site, it attaches to that antigen, which is expressed on the tumor cell, binds to it, and then that tumor cell internalizes the ADC, and then the ADC breaks down, the linker breaks down and releases the toxic payload. That's the basic mechanism of action, if you will. With conventional ADCs, which typically only are targeting one antigen, you reach the limitation of not being able to reach the tumor site. A bulk majority of the ADC actually doesn't even reach the tumor location. It actually stays in the periphery, in the circulation, and then eventually excreted by the body. So that's problem number one, where you have only a small percentage of the ADC that's injected in the body that actually even manages to reach the tumor site. And then once it reaches the tumor site, a proportion of that actually gets internalized, and then you exert the mechanism of action. So that's one limitation. The other limitation related to that is safety. Because most of the ADC actually stays in the periphery, um, the payload, essentially the chemotherapeutic payload or the toxin, eventually finds its way onto the non-tumor locations and sites and healthy cells and exerts its side effect, right? So you have the payload that causes damage to the healthy cells, and that drives majority of the safety or side effects for ADCs. So then you have the safety issue because it's not selective enough and the ADC is not reaching the tumor location enough of it. And so you have all these toxicities build up as well. And part of it is just the construct of these ADCs, how they're designed. And part of it is using older linker technologies which cause premature release of the payload in the periphery, and that exerts a lot of the side effects. So you have essentially challenges both suboptimal efficacy as well as suboptimal safety with conventional ADCs.
NEOK has a bispecific ADC platform. How does it differ from the first generation of ADCs and from other next generation approaches?
Yes. So one, our key differentiation or novelty or innovation is around the antibody itself or the targeting approach itself. So as I mentioned, there are three components of ADC. You have the antibody, you have the linker, and you have the payload. We are not innovating on the payload. So the payload essentially what we are using, it's called the topo-1 payload or topoisomerase payload, which is one of the payloads that has been successful in many tumors. And we are using that similar payload. Where we are truly innovative is on the antibody side or the targeting side. So instead of using an antibody conventionally that targets just one antigen, our antibody targets two antigens or dual targeting. In addition, there is a lot of engineering, sophisticated engineering that's gone into designing these antibodies such that when they bind, they're designed to overcome certain limitations or challenges for the targets that we are pursuing, and we can talk about that in a little bit detail. So it's the approach of you targeting two antigens versus one, and more so also some of the engineering that's gone in really creating this novel antibody constructs. Secondarily, also, we have licensed a next generation linker technology from a company called Synaffix, which is now part of the Lonza group. And it is a fairly clinically validated linker at this point, and one of the top next generation linker technologies, if you will, which allows for overcoming many of the limitations that I previously referenced. It's a very stable linker that you know prevents premature release of the payload, leads to very homogeneous, stable uh, um, ADC production. So there are manufacturing advantages, avoids a lot of the PK or pharmacokinetic issues that we have seen with other ADC. So there are many advantages that this linker technology also brings. So it's a combination of the bispecific approach, the design of these antibodies, the selection of the antigens, as well as the linker payload and the conjugation technology that leads us to a differentiated bispecific ADC construct, which is very unique.
As you think about the therapeutic landscape, what's the gap in the market you see your technology being able to address?
There are many opportunities for the antigens and the targets that we are pursuing. We have two ADCs right now in the clinic in phase 1. NEOK 001, which is targeting B7H3 and ROR1. Those are two antigens. And then NEOK 002, which targets EGFR and MUC1 or MUC1. Between these four antigens or four targets, we can potentially address many different patient population across a variety of solid tumors, primarily lung cancer, head and neck cancer, many GI cancers like colorectal cancer, bladder cancer, certain gynecological cancers. So the list goes on. So we are in the fortunate position that these targets are fairly broadly expressed in many different tumors. And so we will have to see based on our phase 1 experiment, which tumor or which indications that we further pursue. And that will be based on a lot of other additional factors, such as the level of competition, where there's a regulatory path possible, etc. But the potential to address many different patient population across a variety of different solid tumor indication does exist with these assets.
And go a little deeper on the case for dual targeting. What's this able to do biologically that single drug targets can't?
Yeah, so remember when I mentioned earlier that the key goal of achieving better efficacy for an ADC is by improving its localization, meaning ability to actually reach the tumor site, and improving internalization, meaning once it's actually at the tumor site, more of the ADC gets actually internalized by the tumor cell and then eventually is susceptible to the payload. So through a bispecific approach, by targeting of two antigens versus one, you have more likelihood that more of the ADC actually arrives at the tumor site, right? Because you have two antigens potentially that the ADC can bind to. And then once it's there, because of the engineering that's gone into antibodies, what we have seen is that these antibodies are bispecific antibodies tend to internalize much better versus if they were just targeting singular antigen that is included on our bispecific. So, for example, for NEOK 001, which is B7H3 ROR1, we have seen that bispecific antibody internalizes much better versus a antibody that is solely targeting B7H3 or ROR1, and the same is true for NEOK 002. So what we hypothesized and what we have seen in our preclinical data is that these other bispecific ADCs actually have more likelihood to reach the tumor site and get better internalized, and therefore will be able to have more activity, anti-tumor activity and efficacy. Now, on the safety side, it's a little bit of a different case for 001 and 002, and this is based on how these molecules are designed, the bispecific, the format of the antibody, and the type of engineering. But essentially, to simplify a little bit, the NEOK 001, it doesn't necessarily need expression of both the antigens. So we call it an or-gated molecule, meaning it will bind to either of the target antigens when it is expressed. And the reason is that we selected this approach is because both the antigens B7H3 and ROR1 are quite tumor specific. You tend not to find these antigens on healthy cells. And so even if it just doesn't need any both the antigens to actually bind, right? If one antigen is present on a tumor cell, it'll bind. And if both antigens are present, then it'll bind even better and actually differentiate cell and cause even more activity. Now, that is not the case with 002 because the antigens there, EGFR and MUC1, both are actually fairly broadly expressed even on healthy cells. So you want to be careful that by adding another antigen, you have the risk that you will add to toxicity by targeting two antigen versus one antigen. And that's been a limitation with EGFR ADCs as an example, because EGFR-driven toxicity has limited further development of many EGFR ADCs, even though EGFR is a very good ADC target. So the way we designed 002, NEOK 002 is what we call AND gating, which is it requires expression of both the target antigens, EGFR and MUC1, for it to actually bind. Only if one antigen is present, it's less likely to bind because of the fine tuning that we've done with these antibodies. And so, what that actually practically means is that when a healthy cell typically will have either EGFR or MUC1, so it's less likely to bind to the healthy cell, versus only tumor cells are likely to express both EGFR and MUC1 simultaneously. And so that way it becomes even more selective. So you have a case where, in one case with 001, we will get the additional efficacy with the two antigens, but will not add any incremental toxicity because those antigens are very tumor specific. With NEOX 002, we will get the efficacy of EGFR MUC1 without, hopefully, or much less safety issues, because it's a much more selective way of targeting a tumor, because we're going after the dual expression tumors only, and sparing cells, healthy cells, which will express one or of the targeted antigens. That's a pretty long explanation, but essentially that's the principle, that you could exert better efficacy and potentially either not adding incremental safety, or in the other case, be more selective targeting therapy.
You've got two lead programs. You're developing therapies to treat advanced solid tumors with an initial focus on thoracic, gastrointestinal, and gynecologic cancers that co-express the relevant target pairs and are sensitive to topo-1 inhibition. Why start with those indications?
Yeah, for some of the reasons that you described in your question itself, right? There is these tumors, what we've seen tend to be sensitive to topo-1 inhibition. That's an important factor for eventual success of these programs or these molecules. And then the other key factor is the expression profile. These antigens that we discussed are pretty broadly expressed in many of these tumor types and different patient populations. So that's another factor. And obviously, there is high unmet need in all of these indications, right? I mean, where there is currently no topo-1 payload, ADCs, topo-1 based ADCs are still not approved, so it's a pretty open field. And the outcomes for advanced cancer patients in these tumors is still pretty abysmal. So it's a combination of these factors. We are pursuing tumors that we feel have high unmet need, but also where the probability of success is higher. And many of these indications fall in that category.
And what are the remaining technical risks you still need to address for the platform to scale beyond just these few programs?
Yeah, I think I'm not sure if there are technical issues per se. I think one of the key challenges in the field is, you know, what do you do once topo-1 payloads, or payload-based ADCs are approved in majority of the solid tumor indications, which will likely happen. That's part of our vision, that in the next 8 years or so, a vast majority of solid tumor indications will have at least one topo-1 ADC approved. And so the challenge is that once patients receive a topo-1 ADC and progress, they are unlikely to respond to another topo-1 based mechanism of action. So then in that future state, what do you do next? So one of the sort of trends in the field right now is to develop novel payloads beyond the ones that are approved, beyond topo-1 and MMA payload, for example, right? So it's a resistance mechanism. How do you address that? So I would say that is one of the key areas of focus for the field. That is what sort of move beyond the existing validated payloads.
From a business standpoint, what model do you think works best for NEOK? Are you building a wholly owned oncology pipeline, partnering select programs, or using a hybrid approach?
Yeah, we are still a very small company and we have the aspirations to build a sustainable company in the US, starting with these two programs. So we are very well positioned to be to be a clinical stage company right now with two high potential programs across many indications that we can take forward. But we want to maintain the flexibility, that if we have to, so BD and partnering is certainly part of our strategy where we can potentially keep one, partner the other, or develop both simultaneously, but also we have future optionality to get more molecules from our financial and scientific partner and sponsor, ABL Bio, that is already working on many other next gen technologies in the ADC field. So I think we can our vision is to build a sustained enterprise based on the early success of these programs.
You mentioned ABL. You've raised about 105 million primarily from ABL, which is a Korean developer of bispecific antibodies. What's the financing strategy and what's the relationship with ABL?
So to just clarify, we have raised $75 million from ABL as part of the Series A. So they are currently the sole shareholder or institutional shareholder. And ABL, to give you a little bit of a background, is a Korean public company that's traded in the local stock exchange there. It's headquartered in Seoul, and it's a very science-driven company that is, I would characterize it as an antibody company, which has had a fairly good track record of developing numerous bispecific programs or bispecific antibodies for oncology and increasingly for neurodegeneration indications. They have partnered many of their molecules with small companies as well as big pharma, and they also are pursuing their own programs. So it's a very scientifically capable team, an R&D team, and a highly productive R&D team. And the vision for NEOK Bio was to really take their ADC approach and again, leveraging their strength in bispecific antibodies. The idea was to enter the ADC field with true novel innovation and leveraging the strength in bispecific antibodies. They came up with bispecific ADC. So our current vision is to have through NEOK Bio, have a US focused effort, a more focused effort on ADCs, uh, executed through a US team, and eventually attracting US investors, US capital markets, and US talent and expertise to further grow this ADC pipeline through further development and to bring on additional assets. The main financial strategy right now is that, yes, we are quite well funded through our phase one, through the $75 million that we have raised. We should be able to get through phase one and beyond potentially. But our expectation and our plan is to raise additional private financing through a Series B or whatever you want to call it, potentially on the back of our phase one data. And hopefully that happens at some point in 2027, and targeting institutional investors here in the US and beyond. So that's kind of the current financing plan.
There was a lot of interest in ADCs in recent years. Is there a business case to make for bispecific ADC platform to investors as you go out and talk to them?
Yeah, so as you know, ADCs it's a pretty active field, both on the financing side as well as on the BD deal making side. And when you think about, what's next in the ADC space, I mentioned one aspect, which was novel payloads, but certainly that's I would say still a little bit in the future. But bispecific ADC are certainly coming to being. There are many under development in Asia, particularly in China, but not that many in the Western world. We are one of the leading companies that is purely focused on bispecific ADCs here in the US. Particularly with having programs in the clinic already. There are many programs potentially still a little bit early in the preclinical phase. And I think there is a recognition in the field that this is the next wave of innovation in the ADC field, with currently with topo 1 payloads, but with a bispecific or a multispecific approach, which is very novel, combined with the next gen linker technology. So there, I'm seeing you're seeing bispecific ADCs show up on selectively on pharma pipelines now, as well as some smaller companies that are oncology focused or ADC focused. You're seeing selective targets and selective ADCs showing up on some various pipelines. And there are some recent deals as well in this space. As an example, a company called Sidewinder Bio, which is also based in California, announced a Series B financing, and essentially it's a bispecific ADC company like ours, which attracted a fairly good set of U.S. investors than a syndicated Series B. So this is just, I think, points to investor appetite in continuing to invest in this field, and bispecific ADC is one of the sort of next gen approaches that investors seem to be backing.
Given that it is a competitive field, what do you think will allow NEOK to stand out?
Well, it's a combination of our novel approach, right? In the antibodies and the ADC assets itself. The antigens that we have picked are focused on solid tumors. And more importantly, just the timing. I mean, we are, as I mentioned, one of the few companies that are now in the clinic. And so I would say we are in the first wave of these bispecific ADC companies, with potentially data coming up in the next year or so. And so that positions us well from, being ahead of the competition, if you will. And with a choice of targets that are still quite compelling, and with data potentially in the US, right? So that's another factor. Our phase 1 trials are being done in the United States only. And so we will be one of the few companies with phase 1 data from the US that also will favorably position us for the future.
Mayank Gandhi, co-founder and CEO of NEOK Bio. Mayank, thanks so much for your time today.
Thank you. I really enjoyed the questions, and have a nice day.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.